Okay, thank you very much, Ricardo, for the introduction, and also to you and the whole committee for the invitation. That to me that in order to give like this workshop. So as far as I understood, this is mainly for people that want to start, right, with some a new technique in their lab or wherever. So I just try to put some very basic information, some basic steps that you need to follow when you want to do some slice physiology. And that's the reason that the title of this presentation is Slice Physiology, Basic Approach to Study Single Cell Properties and their Synaptic Responses. Because of course, this is part of the thing that I have performed during this postdoctoral experience and also in a previous postdoctoral experience that I also had back in Mexico, where I also studied using in vivo, but also some other in vitro approaches that I will show you later during the presentation. So I want to start this presentation with the definition of electrophysiology, which is a field of research that studies the electrical properties of excitable cells and biological tissues. And this is useful in order to test, to characterize the different properties of these cells. And of course, as we know, the subtle changes in these properties, like these dynamic changes in the properties of these cells, allows the entire system to adapt and to perform the different activities that need to develop. For example, the central nervous system needs to process a lot of information. So these different small changes in the cells that integrate the different circuits that integrate the different areas in the brain are responsible to this. So in order to characterize these properties of the cells and their circuits that they are integrating, the electrophysiology in general terms can divide into different types, which is the in vivo electrophysiology and the in vitro electrophysiology. And the in vivo electrophysiology, of course, as the name says, allow us to use the entire brain, the entire animal, the entire behaving animal, in order, or the patient, depending on the type of study, where we can analyze like different neuronal activity in the brain, and that we can interpret or record it as a local field potentials. That means all these cells working together, firing action potentials together, generating like this oscillatory pattern in the brain. So we can record and study these local field potentials. And also, due to the development of new approaches that are involving like these high density recordings, as we can see it here, and as an example, the silicon proofs, we can also monitor the activity of different cells, recording mainly their extracellularly, recording their action potentials. So we can identify single units or multi-units that are firing different action potentials. And in this particular case, we can start elucidating how this circuit is it's being generated and how it's like important in the generation of different behaviors in the animals. Of course, as we can see here, we can separate in different types of cells or different types of neurons. So we can characterize different things, which not only will allow us to understand how the brain is working in the physiological conditions, let's say in normal conditions, but also we can detect or evaluate the different characteristics that are important to the development of the pathological conditions. So even, however, even when the in vivo electrophysiology has been developing, and it's very important to study like this in the entire animal, the in vitro electrophysiology approach or the techniques have also been developed to detect and manipulate very specific characteristics of these cells. In this particular case, for example, the function of the ion channels, and also we can monitor the action potential generation, and also we can evaluate the synaptic integration of these cells that we are recording. So comparing the different methods, we can see some advantages that probably the in vitro approach electrophysiology will have compared to the in vivo. And the first one is that Now we are working with a simplifier system that we are studying. So we can focus on a smaller number of components. We can control very specific parts of the cell, and we will focus our explanation or the result that we are evaluating based on reduced number of components compared to the in vivo, where we need to consider a lot, even the behavior, right? We can control better the intra and also the extracellular milieu of the cell. And this is particularly important in the pharmacology field, where we want to know how a new drug it's behaving, for example, or it's modulating a response in a cell. Of course, this is also important in order to know the different components, the different neurotransmitters, probably some intrinsic mechanisms, right, in the cell that we want to study. So this will allow us to control a little better like these intra and extracellular areas in the cell, and then use the different techniques. This also allows the determination of the biophysical properties of the cell that we want to record, and this is particularly important because we want to know the very basic alterations in these cells. Also, in the case of the patch clamp recording, we can use current and voltage clamp, which will allow us to identify like different types of recordings that we will see in the next slides. Also, this will allow us the study of responses at a single channel, a neuron, the whole neuron, like the responses in the group of channels and how this will change the potential, the membrane potential of the neuron. And also we can also study the at the network level, for example, the integration of two or more cells that we can patch or we can study in this in vitro approach. We can perform intra and extracellular recording simultaneously, which is something that's probably shared with the in vivo approach. And of course, something that is also important, using in vitro electrophysiology is much cheaper compared to the in vivo approach. And in combination of the in vitro electrophysiology with some other methods, like the proteogenomics or the RNA sequencing, we can generate like very big and important data sets that are more complex, that will allow us to understand better how the cell is being affected, for example, in the development of a pathology. However, we don't need to forget that this also has like some disadvantages compared with the in vivo electrophysiology. And the main one is like the weakness of these methods compared to the other one, which is the fail to completely replicate the conditions of the entire organisms, of course. So this makes complicated to extrapolate totally the result that we are watching in the in vitro approach to the for example, to the behavior of the animal, right? Like in the entire organism. So it's important that we can, I mean, it's always better, and we can see it in these very high impact factor papers, where these two methods, the in vivo and in vitro approaches, are used together in order to describe better a condition. So in general terms, the most used in vitro approach is the patch clamp electrophysiology technique. And this is still the gold standard for studying ion channel. So this is a useful technique to study different tissues and cell configurations. So we can use different types of cells or different types of preparations that we will see in the following slides. And we can assess very small changes in the ion channel's activity, or also we can see it in the entire cell and also in the communication of these cells. So in general, the patch clamp approach was developed in 1976 by these two scientists, Erwin Neher and Bert Sackman, taking into consideration the findings that Hodgkin and Huxley did some years earlier around the 1950s, where they could see, of course, or they studied like these conductances or like these microscopic currents that allow to generate action potentials in the giant axon, in the squid giant axon. So they developed this technique that allow us to record like specific changes in the channels, in the transition of the channels. So this involved using a glass micropipette, touching the membrane of the neuron that we want to record, and then Through this, we can monitor the activity of single channels, like this opening and closing of the channel. We can monitor. So this technique was really important, that these two scientists received the Nobel Prize of Physiology or Medicine in 1991. So in words of the same Sackman, the patch clamp recordings configuration allow us to investigate almost any type of channel in almost every type of cell, which is the main strength of this technique. So the general principle of this technique is the use of a glass micropipette that contains intracellular solution, which is an electrolyte solution that is tightly sealed with the membrane of the cell. So in this particular case, we are isolating the membrane, this membrane patch electrically, and we can monitor the current flow of this, the flow current through these channels, right? So we can, through a differential amplifier, we can record all these cells. So in general terms, the first thing that we need to, or the main principle of these cells, of this protocol, is the generation of a giga-ohm seal, which is created by the attach of the tip of the pipette to the membrane of the cell that we want to record. And in this case, we can approach the cell and then just like touch the membrane, as I have already mentioned. And this is called the cell attach configuration, where we are only touching with a mild suction without the rupture of the membrane. And here we can monitor the activity through specific ionic channels, right? However, as this restricts the access to the cell, to the intracellular milieu, this is avoiding to control the membrane potential of this cell. So in order to monitor and to control more the cell, we need to change we need to use a different configuration, which is called the whole cell recording, where if we apply a stronger pulse or suction, we can rupture the membrane. And in this particular case, we have access to the whole milieu, the intracellular milieu of the cell. There are some other methods that can be used that are not dependent on or having the entire cell, which are called the inside-out recording and the outside-out recording, which consist of, after touching the cell, like in this cell attached recording, we just retract a little the pipette or the recording electrode in order to rupture the membrane. And with some changes in pressure, we can generate like these two configurations. I haven't done it personally, but of course, I mean, these are another option. So in this particular case, we can have the control of the intracellular content of this response that we want to evaluate, compared to the different extracellular milieu that we can change in the artificial cerebrospinal fluid that we are using for as an extracellular solution. So this is important because this allows us also to monitor in a piece of the membrane, just to see the changes and how these can be affected by different pharmacological treatments, for example. So as I mentioned, this method can be applied to different, let's say, to different types of preparation. One of them are the expression system, as the Xenopus oocytes, where we can express specifically some types of channels, and we can, because of this type of method or these cells are very big, we can use two different patch electrodes and monitor not only the current but also the voltage and do different things. So this can be applied to this. Also, in neuronal cultures where we have like these dissociated neuronal primary cultures, we can study like the properties of these cells that are cultured together, probably in this particular case, for example, in the neuronal astrocytes co-cultures, right? We can see the interactions. And also in those cultures that are like an improvement of these last ones, that are called like three-dimensional neuronal models, where they are cultured like layers on over layers of these cultures. And we can have like a better recording because now we have like a 3D dimension and we can study the interaction of these cells. But the most important of all of them is the brain slice models, which represents the most accurate in vitro electrophysiology preparation, as this is still retaining the in vivo-like characteristics of this piece of tissue. So this is, we are getting slices from different areas of the brain. And as you can see, we need to select an area, and then we are studying in these 350 to 450 thickness slices. We can preserve different properties of the circuit without without modifying anything. So this is important, and this is kind of the most used method when we are also using the patch clamp method. So the patch clamp setup, in general terms, is integrated by these different parts, as we can see it here in the cartoon here below. So this is divided in 4 different general parts, which is the environment. That means the means of keeping the preparation healthy. That means all those devices that we are using to keep our slice healthy. For example, an anti-vibration table, a Faraday cage, which will isolate our prep from the external vibration or electrical waves that can be affecting our recording. Also, the use of a specific chamber that allows, for example, have the perfusion of the fluid like on the top and also below the slides. It's really important in order to preserve a little better the activity of the cells that are in our slide that we want to record. And of course, the use of some pumps that will bring the ACSF to the chamber that will allow us to keep the specific conditions to the cell. Of course, we need to have our prep always oxygenated and with a specific temperature control, as we know that the temperature can affect our responses. The optics, which involves all those devices that will help us to visualize the preparation. This is including the microscope per se, which has like different electronic devices, let's say, that will allow us to see better the cell. For example, this DICI infrared contrast. It's always important in order for us to see the difference in the cell, because of course we need to identify where is the membrane and we need to get closer to the cell. So the microscope is a specific, has a specific conditions that will facilitate this. Of course, we need a camera in order to see everything and to monitor everything that we are seeing. The objectives in the microscope will allow us to identify the area that we want to see and later to identify the cell that we want to patch. And of course, the monitor where we are observing everything. The mechanics, in general terms, refers to all those devices that will allow us to put our electrode close to the cell and also perform properly our patch. Of course, this is not. I mean, this is also included as this is the device that we use, this puller that we use for making our specific pipette electrodes, and also the holders of the electrode, and also the micro manipulators, which will allow us to have like a very specific control of the position of our pipette or by the moment that we need to get close to the cell. So, and this is for, of course, this allows us to move like in 3 different orientations, like in all the axes, the X, Y, and the Z axis, in order to have like a good control of everything with a very fine movement. And also, of course, the electronics, which are which are very relevant, that are integrated by the amplifier, I'm sorry, which is the one that is collecting or is recording all the information that comes from the head stage and from directly from our patch pipette, that will collect all this information and also will allow us to manipulate the patch clamp settings, right? Depending on the question that we have. And of course, this is connected with a digitizer, which will change the analog data into digital data, and then will transfer everything, which is subsequently connected to a computer. So of course, the computer needs to run a specific type of software that will allow us to change or to configure these specific methods that we want to use for the different patch approach that we want to have. So in general terms, this is a flow of the electrophysiology approach that we need to follow when we need to patch a cell. So the first step, as we can see it here, is the preparation of the solutions. Is getting ready all that artificial cerebrospinal fluid, right? That we need to use in order to slice our brain, and also to keep the cells, and that we will use for recording the cells. So this includes also the intracellular solutions. So it's better to have the intracellular solutions first prepared and divided in aliquots in order to have a specific amounts of volume, just to not to waste a lot of this intracellular solution. And of course, the extracellular solution has like different contents, right? And in general terms, we are using two different solutions. One solution that is for slicing, and the other one that is for recording. And of course, there are differences in the content of the different salts in these solutions. In order to make for example, the slicing solution, a solution that will protect our slice during the slicing procedure. So in this particular case, we are replacing the high amount of sodium for some other drugs or some other substance. For example, in this particular case, the sucrose, in order to replace the high amount of sodium, which is known that it's the main cause of the alteration of the death of the cells when we are slicing because the passive entrance of sodium will also bring water, and this will increase the probability of the cell to die because of this osmotic change. So there are several methods that are using these salts replacement. For example, the NMDG method, right, which is another chemical in order to substitute the sodium in these cells, in this solution, and try to protect better our cells. Also, another characteristic is that we have an increased concentration of magnesium. So of course, we know that the NMDA receptors work with a zinc ion that is like blocking the magnesium. So we are increasing, let's say, or reducing the probability of these receptors to, I'm sorry, magnesium, not zinc, the magnesium blocker. So we are reducing the ability of these receptors to fire or to be open and cause some excitability of these cells. Also, we need to prepare our slicing equipment that involves the different chambers that we are using in order to hold our slices, the dissecting utensils that we need to use, for example, some scissors, some pliers, and some spatulas, and also a bucket of ice, because it's important always to work with a cold temperature in order to reduce the activity of the cells. And of course, our slicing method, which is the vibraton. So the next step is the proper slicing. So as you can see here in this in this representation in squares, I mean, after properly anesthetizing the animals, we need to get really quick the brain tissue. So we need to open or to retire all the areas, for example, the skin and the bone that is surrounding our brain. Very carefully not to touch the brain and not to damage all the area. And after that, we need to cut or to expose better our area that we want to study. In this particular case, we can see here how they are cutting the cerebellum that they are not using. And for example, for us that we want to use the hippocampus, we are also getting rid of the frontal part of the brain. In that case, we are having like a piece of tissue where we can slice that will contain the hippocampus. This piece of tissue will be glued to the sample plate, and after that, it will be transferred to the vibratom, where we can start slicing our brain. This chamber in the vibratom will be filled with the slicing solution, and we need to keep the oxygenation to the solution and try to keep it cold. And we will get slices from 350 to 450 thickness. And after getting all those slices, probably we can like separate them if we want to study like a specific area and to see if it's like left or right hemisphere. It doesn't matter. I mean, it's only a preparation of the slices. And after that, we need to keep our slices. I mean, we need to keep our slices in a specific under specific conditions in order to help them to recover. In this particular case, the method that we use is after slicing, we transfer them to the same slicing solution at 33 to 34 Celsius degrees for 20 to 30 minutes. And after that, we transfer the slices to the ACSF that we are using for recording for 1 hour. So this will allow all those cells to recover from this process of slicing. So the next step is transferring. I mean, after that period of time, we need to transfer one single slice to our recording chamber, as we can see it here, and with the use of a slice holder, which is like this metal ring with some nylon threads, in order to hold or to give support to the slice and keep it in a specific way, because in a specific position, because we don't want this to move. Otherwise, that will ruin our recording. The next step is loading the electrode into the electrode holder and then slowly approach to the cell. And here, something that is very important, and it's very relevant, is try to identify all those healthy cells from those that are unhealthy, that of course we don't want to patch. So in general terms, we can identify like these healthy neurons if they exhibit like these following features. For example, that they are not shrunken or swollen. They have like a soft contrast in the membrane edges, as we can see it here in this representation. We can properly see where is the edge of the cell, right? And it has like a perfect contrast that we can see here in these micrographs. They have like a smooth membrane appearance. And of course, most of these cells are located a little deeper than the surface of the cell, because of course, when we slice the cell, when we slice the brain, all these slices that are on the surface, of course, are receiving like this mechanical alteration. So there is a higher probability of these cells to be dead by the time that we want to record. So just like a little deeper, we can find like very, very good cells, approximately, or let's say, as it says here, more than 30 micrometers deep. We can find like good cells with these characteristics. Of course, we need to discard all those neurons that exhibit like this crinkled appearance, which are like clearly visible anuclei. I mean, we can see like these very big balloons with another circle in the middle that resemble like a fried egg. So those cells are already dead. So we need to avoid those cells. And also those ones that have like a very dark or very high contrast membrane edges, because these cells probably are damaged, right? So even you can see it, because when you try to patch some of those cells just for fun or something like that, you will see how those cells will explode immediately. After you just touch them or something, you can see how the cell explodes. And of course, you cannot patch anything. So the first approach after you identify your cell is just get your electrode together or very close to the membrane of the cell, and then you will see how the positive pressure that you add in the electrode in order to avoid clogging will generate a dimple in the membrane of the cell. So you just need to get close to the cell, you see the dimple, and of course, if we have like a membrane test monitoring in these cells, we can see our current pulse that we are applying in the electrode, right? After we release the pressure, of course, the membrane goes in close contact to the tip of our electrode, and this will start increasing the resistance in our electrode. As we can see here in the membrane test, how the transitories are small, but now we can see it. Like these transitories means that we are now watching how the current is like probably flowing through the membrane. So of course, as we have, and this is the configuration of the cell attached, only by releasing the pressure or applying a little negative step of pressure will allow us to have like this configuration. But if we don't want to have access to the intracellular milieu of the cell, we need to apply like increased steps of negative pressure in order to break the membrane that it's in close contact to the electrode. Also, the application of some small electrical stimulation will allow us to the rupture of this membrane. So after that, we will see how the transients increase in size, and then that means that we have access to the entire milieu of the cell, and this is called the whole cell recording. And here I am showing you like some links of some very nice videos that I found on YouTube that are very detailed. If, you, for example, you haven't had experience in how to approach the cell and what to see everything, these videos are, I found them like really useful. So I'll just leave them here for you to see them if you want. So of course, the last step is recording of the signal, right? And in order to do this, we have like different methods that we can use in the patch clamp procedure. And for example, we can program all these methods like in the different software that we are using, and we can only record in gap-free mode just to see the spontaneous activity into these cells. And also we can apply or we can inject pulses of current in order to hyperpolarize or depolarize our cell in order to generate action potentials, will allow us to study or to determine the intrinsic properties of these cells. Of course, it's very important all the time to have like a very good recording of everything that you are doing like, with your notes, because some changes can be due to something that probably you didn't do in the next experiment. It's always important to go back to your specific notes. So in general terms, here I will show you an overview of the analysis of the recordings that you can have from the patch clamp techniques, right? And this, as a summary, after we get the slices and we identify our cell and we patch this cell, we can start recording or performing different recordings in order to determine the passive and the active properties of the neuronal membrane. And as we can see it here, we can do different things. As I have already mentioned, when we apply the negative current pulses to the cell, we hyperpolarize the cell, and this will allow us to determine something that it's called the input resistance. When we, which is like the whole resistance of the cell when they reach a steady state. So we can create like an IV plot where we can have the slope of this line that we can create with this response, and that will give you the value of our input resistance. Also, we can analyze the dynamics of the depolarization of these the polarization or the hyperpolarization of these cells, and we can determine some time constant from this type of recordings. Of course, when we apply the positive current steps, we can reach the threshold where the cell will fire an action potential. And after we identify the action potential, we can create like different representation of these. Like for example, this one that it's called the phase plot, which is representing the changes in the membrane potential in time, right? So here we can analyze like different parameters from the action potential. For example, of course, the resting membrane potential, which is the value like before the action potential, the threshold, which is the value where the action potential started, right? The maximum amplitude of the peak, which is like this amplitude or the size, how large is this peak? The time to peak that can be from threshold from the moment that you apply the electrical stimulation. The half width, which is representing the value in amplitude, in time, I'm sorry, the time where the action potential reaches the half, let's say, or the half, or we reach the half of the action potential. And this will be important in order to know the kinetics of the action potential. Of course, there are some other measurements like the maximum rise slope, the maximum decay slope that will evaluate like these differences in the kinetics of the action potential. And of course, after hyperpolarization, which is important to study some other mechanisms that will show or will be representing all alterations maybe of those mechanisms that are important for the repolarizing phase of the action potential. So in order to do this, I will give you like a brief introduction of the work that I have been involved, which is mainly studying the Alzheimer's disease, which is defined as this irreversible and progressive neurodegenerative illness, which is characterized by the loss of the memory and this progressive cognitive capability, which was first described by Dr. Alois Alzheimer when he described all his observations that he did in a patient that he had. And after this patient died, he observed that different morphological changes in her brain, which included like the shrinkage of the tissue, in general terms, the brain, the increase in the ventricles in this and also it includes some or two specific markers, which is the aggregation of two different proteins. One, which is the amyloid beta protein, and the other that it's the tau protein. So in general terms, these are some morphological changes that we can see, but also physiologically speaking, it has been described that in the early phase of the development of the Alzheimer's disease, there is an increase in the excitability of the neurons. As we can see here in this plot, there is a high correlation of the number of seizures that a patient is having with the time that where the neurodegenerative condition was detected. Of course, this has also been studied in animal models of this condition. So we decided to study the hippocampus because it's an important area in the process of learning and memory. And in particular, there is an area in the hippocampus that is called the dentate gyrus, right? Which in general terms, in some other illnesses like epilepsy, this has been studied a lot, and it has been described that some changes in the dentate gyrus are facilitating the generation of seizures. So we used a specific animal model of the Alzheimer's disease that it's called the mouse TG2576, which overexpress a mutant form of the precursor protein of the amyloid beta. And it will have elevated levels of this amyloid beta, and when the animals are old, the generation of the amyloid plaques. So something that is important from this method, that it has a very slow progression of the illness. And as we can see here, in the young animals that we wanted to study, we don't have like these very big aggregates of amyloid beta. So after this, we studied the spontaneous activity in these animals, where we can have, for example, in the current clamp mode or in the voltage clamp mode, where we are evaluating changes in the resting potential of the membrane, and also the currents associated to these changes. So we can characterize the different excitatory or inhibitory currents in these recordings, and we can plot them and compare the two conditions. In our particular case, we saw that in the Alzheimer's disease animal, we have an increased frequency of the excitatory postsynaptic potentials that we were recording, which of course is highlighting the excitability process that these animals have. When we recorded in voltage clamp and we recorded the currents, we see that this is correlated with this increment of the potentials. So also, when we are studying in the voltage clamp, we can also study or analyze the inhibitory currents, right? So in these animals, we see that not only the excitatory currents are increased, but also the frequency of the inhibitory currents is decreased. So using pharmacology, we can see, or we can elucidate like these differences in the different types of currents that we are having. For example, if you hold the cell at different potentials, you can separate from AMPA-mediated currents to those that are NMDA-mediated excitatory currents, of course. So in this particular case, we saw that also the NMDA-mediated currents are increased. So in general terms, in these animals, as you can see here, this is the pharmacology study. And also, for example, you can study the different kinetics of these cells in order to elucidate that these are NMDA-mediated or AMPA-mediated, and then you can quantify that, and then you give like a specific explanation according to the type of current that you're seeing. So in general terms, when we put together all these excitatory and inhibitory currents, we can see that there is an enhanced excitation to these cells, to the granule cells in this animal model of epilepsy, which can explain the hyperexcitability condition. So of course, we can also perform the analysis of the intrinsic properties, as I have already mentioned before. And in this particular case, we were expecting to have alterations that show the excitability, enhanced excitability of these cells. However, when we analyzed our recording, we saw that the resting membrane potential, for example, of the cells, of the granule cells in the transgenic animal were more hyperpolarized, which is a reduce in the excitability of these animals. Same thing with the tau constant and also with the input resistance, which is suggesting that these cells are less prone to fire. So instead of participating in the alteration that give rise to the excitability condition, apparently these cells at these very early stages are trying to compensate for those increased excitatory inputs that these cells are receiving. So just very quick, in 2 minutes, I will tell you that there are some other methods, in vitro methods, that you can use using the patch clamp alternative, where you can analyze or study the synaptic connectivity. And this involves the study, for example, when you patch 2 cells, you can start stimulating one, recording the other one, and then you will see the response of one compared to the other one, right? So, but this not only finish in pair of recordings, but also there are some approaches where they are using several cell recordings, like from 8 to 12, or I guess even more patch clamp recordings, simultaneous patch clamp recordings. And this will allow us to study the interactions in between the different cells and generate like these maps of interacting cells. Of course, this is a method that the Allen Institute has been used in order to create like these maps of neurons that we can now separate when we combine them with the morphological processing or development of these cells, the electrophysiological processing, that we can see that now we can have several types of excitatory cells and several types of inhibitory cells located at different levels in this particular case in the neocortex. There are some other methods that allow us to study very specific parts, for example, in the synaptic transmission. For example, in this paper where there is they were studying the botons and also the soma of these cells, where they can now discriminate between the pre and post synaptic alterations. And of course, there are some other methods that we can patch the membrane of some other subcellular structures like the mitochondria. Of course, this can be in combination with some in vivo methods like the 2 photons, which will give us more information of the cells. And finally, there are some other methods that we can use for performing in vitro recordings in a similar way as the in vivo recording. We can have like these multi-electrode arrays where we can place our slides and then start monitoring the action potentials, and some others where we inject some current and then we can monitor the response of different circuits when we stimulate the afferents of these to these different cells. So I will yeah, I'm not mention more of this. I mean, I just wanted to mention that, for example, in an animal where we injected the amyloid beta and we performed this method, we can see that those animals that were treated with the amyloid beta present a higher excitability, right? And when we study the response, when we change the time in between when we applied the first or the second stimulation, we can determine some specific pre or postsynaptic alterations. So these are here only the reference that I used to put together all this information. And I just want to mention that these are the institutions that have supported all this recording. And with no further ado, I just want to thank your attention. And if there is a question, I mean, or a comment, something that you want to mention about this protocol, I mean, the forum is open. And thank you very much.